Swaranjit is an advisor HP and Punjab government for setting up of colleges and universities. In the 70th Foundation Day of CSIR on the 26th September 2012, two of his publications were in the list of 70 best publications of CSIR based on high citation index. Dr. Swaranjit is a well connected with the different ministries like DSIR, DBT, MOEF, DST, etc. in the state and central government. He evaluated hundreds of PhD, MTech, MSc theses of many institutes and universities and conducted viva voce examinations. Dr. Swaranjit Singh was inducted as a member of the Bhopal Action Plan in and around UCIL last year from CSIR. The Action Plan Remediation Report was released in Bhopal on 23rd August 2013. He is also an advisor, SD College of Pharmacy, Kim Surat 2013. He is the lead person in the brainstorming session in BPU Mohali on future perspective of bioprocessing and compilation of recommendations to DBT 29th August 2013. All right. He is a member of NBA Chennai Task Force in Biodiversity Environment. So with all his achievements and all his experiences, I hope all of you are now excited to listen from him. So now, sir, I would just like you to address the audience. Yes.
Thank you very much for the nice introduction.
Yes, sir. I hope, yes. So everybody is excited just by listening your achievements and experiences. So I hope the audience gets a lot to learn today from you.
I'm very thankful to the St. Xavier's College for giving me this opportunity to interact with you.
Yes, sir.
Especially
Thank you, sir. Yeah.
Dr. A.K. Srivastava, the head of Department Botany, and your principal, sir, and also MSI, who gave me this opportunity to discuss with you something about molecular biology. So we have very good speakers today, and they will cover a lot of molecular biology. I will just give you some overview of what is molecular biology in very simple words. So can I have my first slide? Natasha will project the slide, please. Ria and Natasha, they will project the slide. Dr. Srivastava, are they doing it? Are they?
Ria Gupta, are you listening?
Yes,
Please,
sir. Yes,
yeah, please start it.
Okay.
yes, sir.
When I heard the topic molecular biology, I immediately remembered our Indian American Har Gobind Khorana. He was the first Indian to get Nobel Prize in medicine. Indians are very, very good scientists. And this was 100 years old, when Har Gobind Khorana got the Nobel Prize. That means we have a lot of potential in India to work on molecular biology, microbiology, biochemistry, and so on. Yeah, you can make it full screen. All right. So this is a picture from Science magazine about molecular biology. They have very strong electron microscopes now. They can detect very minute nanoparticles using the electron microscope. We have progressed a lot in the vaccine technology. Next slide, please. Next slide, please. Next slide. Yes, we have progressed a lot in the vaccine technology, in nanotechnology, and the cell over the years. What is molecular biology? We should know the definition. In short, what is molecular biology? Molecular biology is the study of biology at a molecular level. The field overlaps with other areas of biology and chemistry, particularly genetics and biochemistry. Can you please go ahead with the slides because we should go together. Next slide. Next slide. Yes. Molecular biology is a branch of biology that deals with the molecular basis of biological activity. Chiefly concerns itself with understanding and the interactions between the various systems of a cell, including the interactions between the different types of DNA, RNA, and protein biosynthesis, as well as learning how these interactions are regulated. As I told you, I immediately thought about our Indian American who discovered genetic code in India. He is no other than Dr. Professor Hargobind Khorana. Hargobind Khorana is a chemist who cracked DNA's code and made the first artificial gene, was born in poverty 100 years ago in an Indian village. Molecular biology started as early as 1800, starting with inheritance and evolution. In 1928, antibiotics were discovered by the bacteria, by the fungi, by the yeast. 1931, a very good instrument of molecular biology, gel electrophoresis, was developed as early as 1931. So let us see what happened from 1970 to 2020 in the advancement of molecular biology. In 1970s, Boyer and Cohen developed recombinant DNA technology. 77, Gilbert and Sanger devised techniques for DNA sequencing. In 80s, 83, Maulis invented polymerase chain reaction (PCR) process. First disease-associated gene is molecularly mapped to a human chromosome by Husella and Wechsler. In 90, the NIH and Department of Energy launched the Human Genome Project (HGP). 94 Human Genetic Mapping Goal of HGP met. 95 Human Physical Mapping Goal of HGP met. 96 HGP starts testing strategies for large-scale sequencing of the human genome. 97 in what would have come to be called the Bermuda Principle. HGP researchers affirm the rapid public release of genome sequence data without restriction on its use. 2001, HGP International Consortium publishes a first draft on the human genome sequence. In 2002, the International HapMap project is launched to gather data on human genetic variation and could be used to speed identification of genes associated with common disease. In 2012, let us skip some, the Human Heredity and Health in Africa, H3 Africa Consortium, holds its inaugural meeting in Ethiopia. In 2018, TCGA publishes the Pan Cancer Atlas, a collection of cross cancer analyses based on its complete data set. The NIH begins enrollment of All of Us research program, a large scale research resource of more than 1 million participants who will contribute genetic, health, and environmental data. In 2020, first patient with sickle cell disease is treated with an ex vivo approach using CRISPR Cas9. Let us see what are some of the applications of molecular biology. First one is research and development. The second one is diagnosis, paternity testing, pedigree verification, forensic analysis, gene therapy, drug design, and genotyping. I want to just talk to you a little bit about lab on a chip. A chip is a lab. One of the many lab on a chip technologies built in 2020 for disease diagnostic tools is TriSilix. A team of researchers developed this tiny PCR chip made of silicon, powered by a typical smartphone battery. The researchers thought that the simplicity of this technology might help the production of the chips to be relatively cheap and easy. The chip designed for a patient to use at home. It has a DNA sensor, a heater, and a sensitive temperature gauge. Like a tool providing results for blood sugar, glucometer, a small instrument, it measures sugar, and it is known as a glucometer, which tests blood sugar at home. The lab on a chip may also provide quick results for infectious disease. In the future, researchers hope that this device could help people test themselves at the comfort of their home for diseases like cold and flu. Now, what are the advantages of 3D cell culture? 3D cell culture narrows the gap between in vitro and the in vivo systems. Two-dimensional cell culture models are two-dimensional, but tissues and organs are three-dimensional. 3D cell cultures offers more in vivo relevant information. This is all I wanted to tell you about the advances of molecular biology. Now we will listen to very good talks on molecular biology by the 5 participants. Thank you.
Thank you, sir. It was really very informative. Now we would move for the presentation of student speakers. The first student speaker for today's webinar is Ms. Poddar Mayuri Rajendra. She is a second year BSc Biotechnology student from Nauroshchi Wadia College, Pune. She is the president of Microbiology Society of India, Pune Student Unit. She is very keen in participating in activities that enhances one's mind. Today she is going to enlighten us on the topic RNA editing and mRNA transport. Ms. Mayuri.
Thank you, Manisha Pani, for the introduction. Good evening. Myself, Ms. Poddar Mayuri Rajendra, student of BSc Biotechnology, second year. Today's my topic is RNA editing and mRNA transport. I'm sharing my slide. Can you see my presentation?
Yes, Mayuri, we can see your presentation.
Thank you. So, as we know, RNA editing only takes place in eukaryotes and their viruses. RNA editing is a type of RNA processing and it was discovered by Penne and co-workers. In the post-transcriptional modification of an RNA nucleotide sequence, the types are substitutional editing and insertion and deletion editing. RNA editing of either type lead to the formation of transcripts whose sequence differ from that of genome template. This sequence can generated by RNA editing are different from those arising from 5' capping, 3' polyadenylation, and splicing additional RNA processing events that occur during mRNA biogenesis in eukaryotes. Uh, these are the RNA modification enzymes. As given in the diagram, we can see how the transcription processes is taking place and how it gets divided into perfectly matching mRNA and partially matching mRNA. In perfectly matching mRNA, the formation is taking place and it is choked up. And in the second partially matching mRNA, no translation process is taken further. Next, molecular and cellular function of the long non-coding prostate and breast cancer. Long non-coding RNAs are defined as non-coding RNA transcripts with a length greater than 200 nucleotides. Research over the last decades had made a great strides in our understanding of introns, especially in the biology of their role in cancer. In this article, we will briefly discuss the biogenesis and characteristics of introns, then review the molecular and cellular function in cancer by using prostate and breast cancer as example. In the given diagram, you can differentiate between A, B, C, and D, where the whole process is mentioned. As I mentioned earlier, what is splicing? The process by which the introns are removed from the hRNA, that is heterogeneous nuclear RNA, to produce mature messenger RNA that contains only exon. As we can see, cancer immunotherapy. RNA editing is a post-transcriptional process in which nucleotide changes are introduced into a RNA sequence, many of which can thus contribute to proteomic sequence variation. As in the given diagram, the whole cycle is mentioned how the process takes place. Dicer-regulated RNA editing has been found in different types of cancers. The associated epitopes have been identified on MHC molecules and several elect immunoresponse. Several bioinformatics tools identifying RNA editing events in RNA sequence data ready tools was used with RNA sequence data from blood samples from systematic lupus erythematosus patients to identify abnormally high levels of RNA editing. Some may generate novel neoantigens. How the process of mRNA transport takes place. What does it mean? Exonal messenger RNA, that is mRNA, transport serves to the spatially restrict protein synthesis to the exon and growth con. The molecular mechanism of exonal mRNA transport involves the recognition of zip codes by RNA binding proteins, assembly into RNA granules, and transport along the microtubules and actin filaments in the exon and growth con. Local translation of mRNAs provides developing growth cons with the capacity to autonomously regulate the structure and function during the axonal growth and navigation. In mature axons, local protein synthesis is stimulated on injury and might facilitate the process of regeneration. RNA transcription and control of gene expression. In this, we will also able to see gene duplication and speciation. Transfer of RNA is the smallest of the RNA types around 4s because it serves an adapter function for amino acids in protein synthesis. At least one unique tRNA exists for each of the 20 amino acids typically found in protein. Transfer RNA has a significant amount of tertiary structure composed of several loops and stems. Addition to adenine, guanine, cytosine, and uracil, several unique bases such as pseudouracil and dihydrouracil are also found in tRNA. Mushroom poisoning. In mushroom poisoning, we are able to see Amatina phalloides, and this is the most common form of mushroom poisoning in the world. It is the strong inhibitor of RNA polymerase II mRNA synthesis and moderate inhibitor of RNA polymerase III, that is tRNA5 and 5S RNA, and has no effect on RNA polymerase I, that is rRNA. Next, there are some stages of mRNA transcriptions in eukaryotes. There are three RNA polymerase in nucleus, that is RNA polymerase I, that is transcript in rRNA, RNA polymerase II, transcript in mRNA, RNA polymerase III, transcript in tRNA, 5S RNA, and snRNAs. So this is the diagram where you can see the process or where the transcription is being taken place. So in general processes of transcription, what is the basic and general process for transcription is, there are three types, that is initiation, elongation, and termination. So what does initiation do? RNA pol binds to the promoter region, and RNA pol has a sigma factor, which is prokaryote, which recognizes promoter site and initiation transcription. Elongation. In elongation, RNA pol in the presence of magnesium with two positive ions, that is at 2 ATP, catalyze the process of elongation. In termination, when RNA polymerase reaches the terminator region, chain terminating protein RH rho factor stop the synthesis of RNA. So basically, if we are looking for RNA editing, so RNA, as we know, RNA editing only takes place in eukaryote. So for the mRNA transport, the main factor which is responsible is only RNA pol 2. So this is the process where it takes place. That is, RNA pol 2 is created to the DNA by transcription factor, due to which the transcription bubble forms. After the formation, phosphorylation of CBP, that is carbox terminal domain, during the initiation by TF2H factor, helps the polymerase escapes the promoter. Within that, the transcription elongation is added by the elongation factor after TF2E and TF2H disassociates, due to which the termination occurs when CBP of RNA pol 2 dephosphorylated at terminator sequence, due to which termination occurs when poly A sigma comes in mRNA transcript. So basically, RNA transcription in eukaryote, like RNA pol 1 plus RNA pol 2 plus RNA pol 3, the total weightage is 500 kilodalton. So what does RNA pol do? RNA synthesizes 18S, 5.8S, and 20S type. RNA pol 1 is basically a premature RNA where 45S matures, 18S, 5.8S, and 28X. In RNA pol 2, mRNA synthesizes miRNA and siRNA. What is miRNA? It is the microRNA, and siRNA is small interfering, in which it is the most important and advanced in biology, able to regulate the expression of gene. And RNA pol 3 is the tRNA synthesis plus 5S-RRN. So, what is moving forward to post transcriptional modification in eukaryotes? Before this, we will take place in nucleus, but now mRNA has to come out of nucleus, and transcription results in formation of premature mRNA in nucleus. This premature mRNA has to come in cytoplasm, that is, it needs modification. So the modification is being taken place into three parts. The parts are like 5' capping, 3' polyadenylation, and RNA splicing. So what is 5' capping? At the 5' end of mRNA, 7-methyl guanidine is linked, which is 5' and 5' triphosphate linkage. 5' cap helps to protect mRNA and ribonucleus present in cytoplasm. Therefore, it protects from degradation. 3' polyadenylation, in which at the 5' end of mRNA, residues are immediately added to polymerase. And what is in RNA splicing? RNA splicing is the process that removes the intervening non-coding sequence of genes, that is introns, from pre-mRNA and joins to protein coding sequence, that is exons, together in order to enable translation of mRNA into a protein. The further are the images for how does the process is taking place in post-transcription modification. By this, I end my presentation, and the references were taken from Methods of Immunology, RNA 2001, Methods in Immunology 2021, and with the help of Google. Thank you.
Thank you, Ms. Mayuri. Your slides were really informative and at the same time really interesting. The second speaker for today's webinar is Mr. Maruthu Pandian. He is pursuing his first year PG in microbiology at the Gandhigram Rural Institute, Dindigul, Tamil Nadu, and he would speak on the topic transcription in prokaryotes.
Good evening, one and all present here. Thank you, Manisha Pani, for introducing me. I'm Maruthu Pandian A from pursuing first year microbiology in Gandhigram Rural Institute, deemed to be University Dindigul, Tamil Nadu. Now we are going to see about transcription in prokaryotes elaborately, especially in E. coli. I'm just sharing my screen. Mayuri is presenting.
Sorry, just a minute.
Whether my slides are visible?
Yes, your slides are visible.
Thank you. Now we are going to see about transcription. These are the topics I'm going to cover now. Introduction. First, we all know about central dogma of molecular biology. In molecular biology, central dogma illustrates the flow of genetic information from DNA to RNA, RNA to protein. It is defined as the process in which the information in DNA is converted into the functional product. The three processes collectively known as the central dogma, namely replication, transcription, and translation. As the image shows, the DNA is first replicated and then it translated into RNA, followed by RNA into protein via translation. Then transcription. What basically transcription is known as, whether it is prokaryote or eukaryote, transcription is a process by which the information is in a strand of DNA is copied into a new molecule of messenger RNA. Transcription is the first step in gene expression, in which information from a gene is used to construct a functional product such as a protein. The goal of transcription is to make a RNA copy of gene DNA sequence. The steps involved in transcription are initiation, elongation, and then termination. As the picture shows how transcription occurs. Then we are going to see about the difference between the prokaryotic and eukaryotic transcription. The DNA transcription is in prokaryote and eukaryote are different, while the replication and translation are quite similar. The difference in transcription are, the first one is the prokaryotes. In prokaryotes, both transcription and translation takes place in cytoplasm, where in eukaryotes, transcription takes place in nucleus and translation takes place in cytoplasm. This is because bacteria doesn't have the membrane-bound organelles such as the nucleus. And the second one is mRNA processing is absent in prokaryotes. In prokaryote, RNA is the functional unit because they don't have any junk elements in their DNA. Whatever codon is present in DNA, that all codes for specific protein. That is, all of them are exons. Thus, the RNA is transcribed into protein. But in eukaryotes, as previously Mayuri said, the synthesis RNA is prematured RNA. For maturation, the mRNA undergoes splicing, capping, and tailing. And the third difference is coupled transcription and translation takes place in prokaryotes. But in eukaryotes, as I already said in the previous point, the transcribed RNA is immature. After maturation, the mRNA comes out of nucleus and translation takes place in cytoplasm. Then the only one RNA polymerase II enzyme is used for transcription in prokaryotes, while 3 different RNA polymerase enzymes are required for eukaryotic transcription. Okay. Then we move on to the core part, the initiation transcription, prokaryotic transcription. The enzyme RNA polymerase II needs a promoter region to start its work, where the promoter site contains a consensus sequence. Right after the promoter site, the start site is present, which is also termed as the site 0 or site +1. The start site is placed where the transcription actually starts. We can see in the image that start site is denoted in red color. Okay. Then the continuation of initiation, RNA. The nucleotide present before the start site are termed as upstream or downstream. The nucleotide present before the start site are termed as upstream, and the nucleotide present that are present after the start site is termed as downstream. The promoter site, that is the consensus sequence, will be present in the -35 to -10 upstream of the start site, where -35 units nucleotides are TT, GA, CA nucleotides, and -10 are TA, TA, AT. We also know that we are termed as a Tata box. In the Then RNA polymerase II is alone called as core enzyme, where RNA polymerase can bind to any nucleotide of DNA, but it cannot recognize the promoter site, where the promoter site RNA polymerase cannot recognize. For recognizing the promoter site and binding tightly to the DNA, the RNA polymerase II needs a protein factor called as sigma factor. After attaching to the sigma factor, the RNA polymerase II is collectively known as holoenzyme, where before it is termed as core enzyme, after attaching to the sigma factor, it is termed as the holoenzyme. We can see in image that I have explained. When the sigma factor attached with the enzyme RNA polymerase II, the RNA polymerase II stretches itself up to 60 to 80 base pair to find the promoter site where it is. The role of sigma factors are to detect the promoter region properly and to adhere firmly with the DNA strand. Only after the sigma factor releases from the RNA polymerase II, it can start the transcription process. That is, after the strong adhering of RNA polymerase II in promoter site, the sigma factor releases from the RNA polymerase II. And how the signaling works between the RNA polymerase II and sigma factor, and why sigma factor releases from the RNA polymerase are still completely unknown. They are still a puzzle here. The RNA polymerase II and sigma factor forms a complex which binds to the DNA is termed as closed binary complex. And when the sigma factor leaves the RNA polymerase II will back to its original state. Forms a transcription bubble, which is termed as open binary complex. We can see the image. The transcription looks like a transcription bubble, like structure is termed as transcription bubble. Okay. Once the sigma factor releases completely from the RNA polymerase II, the stage is called as ternary complex. Then the RNA polymerase II produce some small stretches of RNA which are not completely functional sequences. They are just termed as abortive initiation of transcription. They are small stretches of RNA which does not code for any protein. They are just called as abortive initiation of transcription. After the abortive initiation transcription, the initiation for synthesis of complete mRNA starts, which is followed by the elongation process. Then we move on to the elongation process. It is the actual phase for RNA polymerization. That means the ribonucleotides are added to the sequence one after another to produce a long chain of RNA. That the RNA is called as messenger RNA because it carry all the sequences and codon which codes for protein. The required ribonucleotides are present in cytosol of the prokaryotic cell, where it is present in the nucleus of the eukaryotic cell. Because prokaryotic cell doesn't have the membrane-bound organs, no. That's why all the ribonucleotides are present in the cytosol. Elongation always done by RNA polymerase II on the 5' to 3' direction of newly synthesized RNA strand. That means the coding strand will be 3' to 5'. The particular protein called bridge helps RNA polymerase II in adding the nucleotides. Okay. The DNTPs are AUGC. Then the protein bridge is a type of protein subunit protein factor which later associate with RNA polymerase II. The role of protein bridge is to free only one nucleotide base pair from the template strand to react with the RNA polymerase II. Because if one or more nucleotides releases from the template strand, then it will cause mutation. And the second one is ensure only one ribonucleotide binds to the template strand at once. Okay. Then the bridge protein cannot ensure whether nucleotide pairing follows base pairing rules, whether RNA polymerase II itself responsible for proofreading activity. The bridge just confirm that only one nucleotide pairs with the template strand, whether it will not follow the base pairing rules. But RNA polymerase II have the proofreading activity which checks the correct nucleotide pairs with the template strand. If any wrong nucleotide is added with the RNA polymerase, then RNA polymerase uses its exonuclease activity by which they cleave and take out the wrong base pair and add the correct one. The proofreading activity of RNA polymerase is not good as DNA polymerase, where DNA polymerase proofreading is very accurate than the RNA polymerase. Okay, then we move on to the termination. The prokaryotic termination, transcriptional termination of two types. Intrinsic termination and row-dependent termination. Intrinsic termination is also known as row-independent termination. First, we are going to see about intrinsic termination. It is a sequence-specific termination which forms a hairpin loop. The RNA internal sequence have more GC-rich palindromic content. The GC hairpin structure is followed by polyuse at the end of the RNA. It is because the termination sequence of DNA contains an adenylation, polyadenylation. That means on the A in the DNA, which is followed by the RNA, which binds to the U, poly-U, which forms the poly-U adenylation in the end of the sequence. The hairpin loop is very strong bond because of the GC binded with the triple hydrogen bond, where AU is the weak bond when comparing with, which is bound by a 2 hydrogen bond. It is a weak bond when comparing with the GC bond. Okay. The difference in the interaction actually drag the RNA out from the DNA segment, which results in dissociation of the RNA from DNA. Because G and C binded with the 3 hydrogen bond and A and U bind with the 2 hydrogen bond, there will be the force interaction differences. Because of this, the RNA dragged out from the DNA, which results in a mRNA, complete mRNA out. These are the image where we can see GC-rich hairpin loop. Then row-dependent termination. Row-dependent termination is involving of different enzyme called row units. Row enzyme are nothing but helicase enzyme with 6 subunits. We all know about the helicase enzyme in the replication. It helps to the unwind. Like that, it is the same enzyme with 6 subunits used in the row-dependent termination. The lot of cytosine-rich region present in the RNA, which acts as a signal for binding of row protein. And the site is termed as red site or red site. The red means for the row utilization site. In red site, the content of C will be more than 41%, where the content of G will be less than 5%. Okay, that way it is known as lot of cytosine-rich site. Then the characteristics is row protein. Row protein is a hexameric protein which has only 2 different domains, N-terminal domain and C-terminal domain. Row protein is a rotating enzyme where it drags energy from ATP hydrolysis for rotating. It rotates from down to high by which means of hydrolysis. ATP hydrolysis, it gains energy. By using the energy, it rotates from up to down. Rho can separate two strands, whether it can be DNA or DNA RNA. Rho binds to the red site and migrates toward the termination site. We all know there is a termination site in the DNA where the termination must happen. When the RNA polymerase reaches the termination site, the row binds to the red site and moves towards the termination site. Once it reaches and ended, it passes it between the DNA and RNA bond. It passes through the DNA and RNA binding and removes the RNA from the template. Thus, results in the DNA releases from the RNA, the results with the functional mRNA alone from the DNA. Okay. Then this is the overall transcription in prokaryotes, where initiation has the sigma factor with binds with the RNA polymerase and initiate the transcription. And after the elongation is done by sigma factor. After the removal of sigma factor, the protein, bridge protein adds the nucleotides. Then the termination is added by the row dependent or row independent. Then after the termination, the resultant mRNA with the exon, which are all codes for the protein. Okay. Then these are the recent research in transcription in E. coli, or that means the prokaryotes. Rho is a general transcription termination factor in bacteria. Here, the use of single molecule fluorescence assay to provide evidence that Rho-mediated transcription termination can occur via three kinetically different routes. There are three different routes that are going to be proved where the Rho is traveling to enhance the transcription termination factor. Then the signaling work between the sigma factor and RNA polymerase 2 are completely persist, where the works are going to sense the signal. We already seen the sigma factor releases from the RNA polymerase when it reaches the promoter site. The signal between the RNA polymerase and sigma are still unknown. The research are going to sense the signal. The transcriptomics differentiate two novel bioactive strains of Penibacillus species, isolated from the perennial ryegrass field microbiome. Okay, these are my references. And thank you. I really thank Microbiology Society and St. Xavier's College Ranchi for giving me this wonderful opportunity. Thank you.
Thank you for an excellent presentation, Mr. Maruthu. Now, moving ahead with our third student speaker for the evening, Ms. Aishwarya Sirshulla. She is a PG student of Institute of Science, which comes under the affiliation of Homi Bhabha State University, Mumbai. She was a COVID volunteer for more than 2 years. She's participated in Micro Olympiad competition, and today she is going to speak on the topic concept of split genes.
Thank you for the introduction. I will be presenting my slide. Is my slide visible to all of you?
Yes, it is.
So, myself, Aishwarya Sursula. I am a microbiology student of Institute of Science, and I will be presenting on the topic split gene, the concept of intron and exon. So, split genes are also called as interrupting gene, which consists of introns and exons, in which introns are eliminated by a complex molecular structure called the spliceosome. I will be covering the following outlines. That is, what are exons its properties, then what are introns its properties, then there are two intron hypotheses, then how these introns influence in eukaryotic gene expression. Finally, I conclude my presentation and the references by which I have made this presentation. As I earlier said, gene consists of introns and exons. Introns are always surrounded by two exons. Now, what are exons? Exons are nucleotide sequences in DNA and RNA that are conserved in creation of mature RNA, by which transcription process begins. The process by which DNA is used as a template to create mRNA. Then further transcription, translation process begins, where this mature mRNA works in conjunction with ribosomes and transferred RNA, which are both present in cytoplasm, create proteins. Now, the characteristics of exons has these codes for synthesis of protein. Exons are the DNA segment which encode a part of amino acid sequence of a complete protein. They are found in both eukaryotes and prokaryotes. They leave the nucleus to the cytoplasm after the production of mature mRNA immediately. They are highly conserved sequence. Exons usually contain both the 5' and 3' untranslated region of mRNA, which are used in transcription. Then, what are introns? Introns was discovered by Richard Robert, where he find discontinuous sequences. So, introns are nucleotide sequences in DNA and RNA that do not code for proteins. Also, they are called as non-coding sequences and are removed during precursor mRNA, pre-mRNA stage of maturation of mRNA by a process called RNA splicing. The word intron is derived from the word intragenic region. They are also called as intervening sequence because there are always presence in between two exon molecules. Like this. Then the characteristics of introns. They belong to non-coding DNA because they don't encode for any amino acid. They are considered as the bases located between two exons. They are only found in eukaryotes. There are two hypotheses by which the reason between why they are not found in eukaryotes I will discuss in further slides. They stay in the nucleus by splicing out from the mRNA primary transcript during mRNA processing inside the nucleus. They are less conserved sequence as compared to exon. They range in size from 10 base pair to 1000 base pairs. Now there are different types of introns. Spliceosomal introns, introns in coding genes. They are also called as pre-mRNA introns. For the splicing, they require the presence of snRNA and other proteins. Then comes the tRNA introns. These introns are basically mainly present in the tRNA genes. Uh, they require enzymes for splicing of these introns, and these enzymes are called also called as special enzymes. These enzymes also cut the RNA molecule. Then the group 1 introns. These introns are present in mRNA, tRNA, and rRNA of chloroplasts and mitochondria. They are self-splicing introns, that is, they don't require enzymes or protein like that in spliceosomal introns and tRNA introns. They can catalyze their own removal from mRNA, tRNA, and rRNA precursors using guanosine 5 triphosphate energy molecule. Then comes the group 2 introns. They are present in chloroplasts and mitochondria. They are also function as group 1 introns. So, in this, they do not require GTP in order to remove themselves. This intron fold itself to form secondary large structure, lariat structure, which fold itself. Then there are two hypotheses for why they are not present in prokaryote introns. So introns, the first one is the early intron hypothesis. Introns were an essential feature of the earliest organism. They were absent in bacteria because of the shorter division time of bacterial cell. Bacteria have many more growth cycle in which to evolve. This evolution has brought about the loss of nearly all ancestral introns. So by the early intron hypothesis, that they say that the intron was present in the primates, but during the evolution, the ancestral introns got lost. The next hypothesis is late intron hypothesis. In this hypothesis, early organism did not contain introns. Introns are relatively recent arrival in the eukaryotic lineage that help to generate a diversity of regulatory mechanisms that are required to control gene expression in multicellular high differentiated organisms. They are considered as a transposable element. In this view, prokaryotes do not have introns because they never had them in the first place. So both these hypotheses are partly accepted. Now the distribution of introns. The intron distribution and 5' untranslated region and the coding region and 3' untranslated region are different for the same organism. While these rules are common for human, mouse, rat, and Arabidopsis and fruit fly. Now, how these introns influence eukaryotic gene expression? Introns are called as junk DNA or selfish DNA. They affect transcription by acting as repositories for transcription regulatory elements such as enhancer or repressor. Introns are also required for specific modification of some exons by, like in process of RNA editing, interaction between pre-mRNA processing events. Also, the nuclear cap binding complex promotes the excision of 5' intron, whereas the interaction between the spliceosome and polyadenylation machinery promotes the excision of 3' intron. This results in the proper 3' end formation, which is required essentially for the transcription process to begin. So this is the summary where the introns get spliced out and the exons get joined. These exons are called mature RNA, which further gets into the process of transcription and then translation of to form protein molecules. So the conclusion: a gene is a segment of DNA which yields a functional protein, either a polypeptide by transcription, by translation process, or an RNA. The intergenic regions of a gene are composed of introns, and the coding region composed of exons. In eukaryotes, coding region structure which split into segments called exons. Introns can be found always between two exons. Introns belong to non-coding DNA. All exons along with intergenic regions are transcribed by RNA polymerase into primary transcript of mRNA. Introns are removed by the primary transcript during mRNA processing called as splicing. Thus, a mature mRNA only consists of exon which codes for uh, transcription and then translation to form protein synthesis. These are my references. Thank you.
Thank you, Miss Aishwarya, for such a wonderful talk. Now I would request a professor from St. Xavier's College, Ranchi, Department of Botany, Mrs. Aroma Aishwarya Barla, ma'am, to take it over. Aroma, ma'am?
Am I audible?
Yes,
Hello?
ma'am, you are. Yes,
Hello?
ma'am.
I'm audible? Okay.
You are
Thank you,
audible.
Manisha. And a very good evening to all of you. And a very warm welcome as well. I feel privileged to introduce our first speaker, Dr. Tanushri from Cactus Communications. Dr. Tanushri has completed her BSc in Biotech from BHU and MSc in Plant Biotech from Terry School of Advanced Studies, New Delhi. Being a major in plant biotech, she pursued her PhD from Terry itself. She has various skills and interests, such as genomic research, PCR, bioinformatics, development of transgenic plants. Currently, she works as a scientific writer and reviewer or editor on contract in Cactus Communications. She has edited over 100 manuscripts in life sciences for journal publications. She has generated experimental evidences of regulatory diversification of conserved gene copies. She has also worked in bioinformatics, and she has also worked in preparing a syllabus for biotech students. With all of this, she has ma'am has attended a lot of conferences and workshops, and she is here to share her experience with her. With this, I would like to welcome Dr. Tanushri to enlighten us on the topic use of lac-operon and alpha complementation in gene cloning. Ma'am, the stage is all yours. Thank you.
Thank you for such a warm welcome, and thank you, Microbiology Society, for giving me an opportunity to share whatever 2 cents of knowledge I have regarding this topic. So, shall I share my screen now?
Yes, ma'am.
Please let me know when is it. Is it visible? Is my screen visible?
Uh, I think, ma'am, it's showing that you are presenting, but your screen is yet not visible. The presentation is still not
Okay,
visible.
let's give it a minute. Okay.
Yes, ma'am.
Okay.
Yes, ma'am. Now your screen is visible.
Can you see my presentation? Okay. Present visible?
Ma'am, we can see the Google Meet screen. Yes, the presentation is visible now.
Okay. And am I audible?
Okay.
Am I clearly audible?
Yes, ma'am, you're clearly audible.
Okay. So should we start?
Yes, ma'am. We may.
Okay. Okay. So hello to everyone once again. And today I'll be discussing two elegant blue-white screening systems which are extensively used in plant functional genomics these days. And for both these systems, we have E. coli to thank for because they are basically assays based on E. coli derived enzymes. So before starting to the screening part, I'll just explain in brief what functional genomics in plants entails basically. So functional genomics means associating genes with functions, and it is generally carried out by performing functional characterization of genes. So the genes are characterized for their function through through either making gain of function mutants or by loss of function mutants. And with mutants in plants, we mean transgenic plants. So transgenic plants are basically mutants with either with an altered function of a gene, so that we can assess the phenotype and determine that what the genes is how the gene is functioning. And this strategy is not limited to protein coding genes only. It works for both non-coding genes like microRNAs, and even characterization of promoters can be carried out using the same strategy. So the strategy of functional characterization basically entails first isolation of the gene of interest, which is then subsequently cloned in a suitable vector where it is maintained and sequence characterized. And after that, it is transferred to the plant using various methods, the most common of which is agrobacterium mediated transformation. And after the gene of interest and the entire expression cassette has been transformed into the plant genome, the plant is then the transgenic plant thus produced is then genotyped for confirmation of the presence of gene of interest, and then phenotype to analyze what the gene is, how the gene is now functioning in its altered environment. So in this strategy, there are two levels at which the blue-white screening systems, which were derived from E. coli enzymes, come into play. And the first cloning. While we are cloning the gene in a suitable vector, then one of the system comes into place then, and the other is while phenotyping. I'll discuss both in my presentation one by one. So the research problem that we had undertaken during my doctoral thesis basically entailed the sequence and functional characterization of three promoters which belong to three homologous genes in Brassica. So the gene that we selected for this purpose was called SOC1. SOC1 is a transcription factor which is responsible for determining the precise flowering time of angiosperms. And the genomic background that we chose was Brassica gentiana, which is the common mustard. And the interesting thing was that the SOC1 gene was present in 3 identical copies within the Brassica gentian genome. But even though the proteins encoded by the genes were identical, their expression patterns varied vastly. So, because the gene expression is driven by their respective promoters or regulatory regions, so we decided to analyze the regulatory regions to find out where the diversification is being rooted from. So, to do that, we first isolated and cloned the promoters, the help of available Brassica sequence genomes, and by preparing homolog specific common cloning pipeline. So the vector used for this purpose was PW1398, which is specifically designed to characterize promoters. Is it hello? Audible?
Hello, good evening, ma'am.
Hello?
Ma'am, actually,
Huh?
your voice was. Hello, ma'am, your voice was cracking a bit. Can you
Huh?
थोड़ा सा explain थोड़ा सा before what you're explaining? Like maybe
कहाँ से?
2 minutes back what you were
From where?
sharing with us.
Okay.
Ma'am, I think
Okay.
in the
I'll start from
previous
here itself.
yeah.
Okay. Okay, I'll briefly explain it once more. Am I audible now? Hello?
Yes, ma'am, you are. Yes, ma'am. Hello, ma'am.
Is
Yes,
my
ma'am.
voice faltering
You're
right
audible.
now? Okay. Okay, so
A
I was
bit.
just
It is
telling that,
faltering a bit.
okay. Okay, I'll continue, and if it's problematic, you can let me know again.
Yes, ma'am.
Okay. So I was telling that functional genomics basically means associating genes with their respective functions, which is carried out through functional is basically entails generation of transgenic plants which carry a gain of function or a loss of function mutant relating to that gene. So that if the gene is overexpressed or the gene is knocked out, so we can see what phenotype changes that is bringing in the transgenic plant. So, and this strategy is not restricted to protein coding genes only, but to microRNAs also and to other functional regions of the genome like promoters. So, functional characterization entails first the isolation of the gene of interest, its cloning and amplification in a suitable bacterial host, which simplifies the sequence characterization process. And once the sequencing is done and the sequence is analyzed, the isolated gene can be transformed into the plant using several methods. One of the most common of which is agrobacterium mediated transformation. After the transgenic plants is thus made, it is the plants are then genotyped for the presence of gene of interest, and then they are subsequently phenotyped to see what kind of changes the altered gene expression cassette is bringing. So in this whole strategy, there are 2 steps where the E. coli based enzymes come into play. First is while the gene is being cloned in the vector, and second is when the plants are being phenotyped. We will discuss both of them in detail. So the research we undertook, the plant functional research brassica. Gene that we selected for research was SOC1, which is a transcription factor that determines flowering time in angiosperms. So this SOC1 is present as three copies in Brassica gentian genome itself, and the proteins encoded by these copies are highly sensitive. So that would mean that perform a similar function, but different. So promoter or regulatory regions, we then try to test the sequence and pattern. So to do first isolated and cloned the three promoters from Brassica and use the already available Brassica sequence genomes, and we clone them in a suitable vector for the characterization. Is it okay? Did you hear till now? Hello?
Yes, ma'am.
Hello?
Yes,
Okay.
ma'am. Yes, ma'am. Yes, ma'am.
Okay. Okay, okay, I will continue. Okay, so the vector that we chose for this purpose was a commercial vector P Cambia 1391Z, which is specifically designed for promoter characterization. It is a 12 kb long vector. It is a 12 kb long binary vector, which is meant for agrobacterium mediated transformation. And it is a shuttle vector. That means that it can replicate both within E. coli as well as in agrobacterium. And the selectable marker for E. coli is kanamycin, whereas the selectable marker when the region has been transferred to plants is hygromycin resistance. The most important feature of this vector is that the location of the multiple cloning site is present within the LAC-Z alpha region, which I'll explain in the next slides. And it is placed such that the inserted promoter will be placed directly upstream to a reporter gene, which is called GUS, which I'll explain in later slides. Both these features will be discussed in later slides. So let's move on. So before that, before discussing the LAC-Z feature, we must discuss about the bacterial host. So the host that we used for our cloning was the common commercial strain E. coli DH5 alpha, and because it permits high efficiency transformation, and it allows stable amplification of DNA and very high yield of plasmid because of the mutations that it consists, like the RAK1 and the A1 mutation. It does not carry any natural antibiotic resistance, and the most important feature is that the LAC-Z gene, the LAC-Z alpha gene, has a mutation which, because of which it enables blue-white screening of recombinants, which we'll explain now. So what is LAC-Z gene? So it encodes for the LAC-Z gene of E. coli basically encodes for a very important metabolic enzyme, which is beta-galactosidase enzyme. This enzyme is responsible for metabolizing lactose into its monosaccharide units, glucose and galactose. And it is, as already mentioned, it is encoded by the LAC-Z of the LAC-Z gene, which is a part of the LAC operon, which is collectively regulated through a regulatory module. I think my next speaker will be in a better position to explain this, but I'll just state in short what it entails. So in presence of the inducer, the operator of the LAC operon is free, and the transcription machinery binds to the transcription machinery and RNA pol binds to the inducer and binds to the operator and allows the transcription of these three genes, which belongs to the LAC operon, the first of which is the beta-galactosidase gene. Also, I must say that the beta-galactosidase gene is composed of two subunits, the alpha subunit and the omega subunit, both of which are necessary for its complete function. So LAC-Z alpha and LAC-Z omega, both subunits are required for its complete function. Now, in the natural setting, the substrate is lactose. The substrate for this enzyme, the natural substrate for this enzyme is lactose, which is and it is induced in the present. Its expression is induced in the presence of a natural inducer allolactose, and the products are glucose and galactose. The assay that has been built around it has replaced the natural substrate with a chromogenic substrate, which is called XGAL, and the artificial inducer is a synthetic molecule, which is IPTG. In presence of both of these, this substrate and this inducer, the product is a colored blue pigment, which is formed after dimerization. So this property of LAC-Z is being used to screen out between recombinant vectors and non-recombinant vectors. How? I'll explain now. So after insertion, After the gene has been isolated, the E. coli DH5 alpha cells were made chemically competent for transformation, and the Pcambia 1391 Z vector was linearized or restricted using a blunted enzyme SMAV1. So after the vector has been cleaved using SMAV1 enzyme, the insert or the gene of interest or the promoter that we had isolated was ligated into it to generate recombinant plasmids, and this ligation mixture was transformed into competent E. coli DH5 alpha cells using the heat shock mediated transformation. So the process by which this blue-white screening will be carried out to differentiate between recombinant and non-recombinant is called alpha complementation. And I'll just briefly explain what it means. So as I had mentioned earlier, that the E. coli DH host does not has an Z alpha that and it from the Z omega and also Z alpha peptide. So what happens is that it cannot produce a functional beta galactoside enzyme. So in presence of the X-gal and IPTG in the medium, cannot produce any pigment. However, when a plasmid like Pcambia is transformed within that DH5 alpha, the function of LAC-Z alpha is complemented from here because it contains so when this function is complemented, such transformed E. coli can then produce functional beta galactosidase enzymes and therefore metabolize X-gal to give blue coloration to the colonies. Okay, this is one step. And but after that, when the plasmid vector has the insert, when the promoter or the gene has been cloned in plasmid, remember the has been built such that it is a lax alpha gene. Come or the gene will be inserted here. Lax gene will be so this will lead to non-functionality of the beta glucosidase because now the laxate or the complementary laxate alpha also non-functional. So the recombinant plasmids which are carrying insert will also unable to produce blue coloration of colonies. This is called alpha complementation. So in a nutshell, what happens? The transformation mixture will contain three kinds of cells. The untransformed DH5 alpha, which were able to uptake any kind of plasmid. Then the equal DH5 alpha cells, which had uptaken plasmids, empty ones, because when you ligate, there were several which would have recircularized without insert in them. So such plasmids will have been uptaken by some cells. So these are called non-recombinant transformants. Then there will be E. coli DH5 alpha cells, which were transformed with indeed recombinant vectors. These are recombinant transformants, and these are the desired product that we want. So when these three cells are plated on LB, or they are allowed to grow on LB medium, which is supplemented with XGAL and IPTG, so the untransformed cells have no functional copy of laxid alpha, so they are unable to metabolize XGAL. So there will be no pigment formation in them. But in these, the second so the result will be formation of blue pigment. And again, in these and in the desired ones, also the complemented laxid alpha has also been disrupted, and therefore there will be no pigment formation. So the two kinds of recombinants can be screened on the basis of their color. However, how will you differentiate between the untransformed and the transformed recombinants, because both will be white? So then selection comes into place. So selection means when you are differentiating between two kinds of cells and one of them is being killed, only one of them is allowed to survive. So when you add kanamycin to the medium, which is the antibiotic selection, the untransformed cells will be unable to survive, and only the transformants will be able to survive the differentiation of color. So this is how blue-white screening for cloning works. So it enhances the speed of the process or the whole cloning process, because now you have to screen, because now you can see that only the white colonies will be recombinant. So you can only pick the white ones and check, confirm them through colony PCR. Otherwise, if the whole plate had been white, full of, you had to screen hundreds of colonies just to find a few transformants. So the efficiency of the whole cloning process is increased many-fold. But all researchers should be aware that this process is prone to false positives and false negatives, because false negatives are mostly caused due to short insert length, or when the insertion has occurred in frame, or there is leaked expression. And false positives generally occur when there is insufficient or poor quality of IPTG or XGAL in the medium, or when there is a suboptimal concentration or quality of antibiotic in the medium, which will allow the untransformed cells to appear, which are also white in color. So that might appear as false positives. So this was one of the blue-white screening assays that I wanted to discuss. Now I'll move on to the next. So in the process of characterization of these promoters, the next steps were, after confirmation through colony PCR, the recombinant plasmids were sent for sequencing, and the sequence characterization and in-depth sequence analysis was carried out on them. And based on those results, 3 representative promoters were selected for functional characterization, which were then transformed into Brassica gentiana through agrobacterium-mediated transformation. Now, let me just introduce you how the SOC1 promoter was cloned. So the part of the DNA, part of the vector that was introgressed into the plant genome through agrobacterium mediated transformation consisted of, first, the antibiotic selection gene, that is the hygromycin resistance gene, hygromycin phosphotransferase 2. Then there was the Brassica SOC1 promoter, which we inserted in the LAGZ alpha frame, and it was placed such that it was immediately upstream of the E. coli GUS-A gene, which is our next reporter that we want to discuss. So this asset is called promoter reporter construct, and this process is called promoter tagging or transcriptional fusion of promoters. So the idea is that wherever the promoter is capable of expressing, it will now express this gene instead of its native gene. So when we performed the phenotyping of Brassica gentiana transgenic plants, they appeared no different from the wild type plants, and were confirmed for the genotype by amplification of the GUS gene in their genome. And now we'll move on to what the GUS gene stands for. So GUS is a gene which was derived from E. coli, which encodes the beta glucuronidase enzyme. It was earlier known as UID-A, and now it's known as GUS-A. And it performs the hydrolysis of beta conjugated glucuronides in the natural system. The best part of this is that it is not native to any of the plants. That means there will be no background expression. So it can be easily used for phenotyping in several in whole the plant. So it is very versatile in terms of substrate. Several synthetic substrates have been found which enable the spectrophotometric, fluorometric, and histochemical assays. And it allows gene tagging, protein tagging, as well as promoter tagging. So it has an array of uses. So in the assay, the substrate used is called X-gluc, and X-gluc is hydrolyzed by this enzyme to again produce an insoluble dimerized product, which is blue in color. So like the black assay, like the beta-galactosidase assay, again, this is producing a blue color wherever it is getting expressed. And by histochemical, we mean that the tissues, the plant tissues, can be directly subjected to this assay to visualize in real time how the expression of promoters is taking place. So This is carried out by preparing a buffer solution, a staining buffer solution, which contains X-gluc and a redox system because oxidization is required for the chemical reaction to complete. And then the tissues are directly immersed in this buffer to stain, and the stained tissues are then washed with 70% ethanol to remove whatever pigments it consisted, like chlorophyll, etc. it consisted before. So the results somehow look like this. So here we have generated a promoter reporter construct of this particular promoter. Sorry, this particular promoter, and as we can see, it is we could find that this promoter was driving expression across several domains, across many domains, vegetative as well as reproductive, and it could clearly differentiate between the floral whorls where the gene was getting expressed and where it wasn't. So this is another blue-white screening assay that we have used. And in contrast, I've shown we made several transgenics, but I've shown only two. So in the other in the next promoter, in the case of the next promoter, we were able to see that this promoter was completely non-functional, and it could not drive Gus expression across any of the expression domains of the transgenic plants. So in this way, we were able to understand the basis why the expression patterns of SOC1 copies, identical SOC1 copies, were varying in Brassica gentiana because of the regulatory diversification that was happening in the promoters. Not only this, we also tested different portions of the promoter for their function, that which parts of the promoter are more, which are less functional. I have taken two panels, for example, like when we took this 2 kb full 1.5 kb full length promoter, it was able to drive Gus expression across several whorls and even in leaves. However, when a shorter proximal fragment was used, it completely failed to drive any reporter expression. That means that, and we were able to pinpoint which portions of the promoters were more functional than the others. So this way, we used this blue-white Gus assay to differentiate, to functionally characterize our promoters and to pinpoint the functional regulatory regions within each of the three promoters. So the Gus reporter system is not limited to histochemical assays, and you can one can use it for studying the anatomy of transgenic tissues if there is some kind of variation and at what level the genes are being differentially expressed. And by making translational fusions or protein-protein fusions, here we prepared promoter reporter fusions, you could make protein reporter fusions, and you could follow the subcellular localization. And the strength of promoters can also be compared by using different substrates. As I told you, it is very versatile in terms of substrate. So you can perform spectrophotometric assays or fluorometric assays or even quantitative real-time PCR to compare the strength of promoters. So this way, we made use of two E. coli derived enzymes and the assays built around them to sequence characterize as well as functionally characterize the promoters from Brassica gentiana in the functional genomics background. So that is all for my presentation today, and I'll be happy to take any questions if anyone has any doubts.
Thank you, ma'am, for your wonderful presentation. And thank you for enlightening us with this topic. Do we have some questions here? If someone has any question and you are not able to share it, you can type it in the chat box so that it can be discussed over here. Hello? Okay. Anyone? Any student? Ma'am, can you again? I have some doubt, and I have Can you
Yeah,
explain a
yeah,
bit
sure.
more about gas reporter system?
Okay.
Actually, there was a
You
bit of
mean the
glitch,
enzyme
so
or
I
just
missed out. I'm sorry.
okay?
Mm,
Okay. I'll
yes,
go
ma'am.
back.
Please.
Okay. So basically, the gas reporter system is based on an E. coli derived enzyme, which is beta glucuronidase enzyme, and it has a
Okay.
natural substrate and a natural product, but it is built such that the assay is such that they have taken a synthetic substrate called X-gluc, which imitates its natural substrate and produces a product which is colored, which is blue in color. So wherever, if we tag if we tag the E. coli Gus A gene with our promoter, so wherever the promoter will be expressed, instead of its native gene, the Gus gene will be expressed. So, and in whatever tissues the Gus gene is being expressed, when you stain it with the Gus buffer, which contains the synthetic substrate, blue coloration will be produced. So all the tissues that are expressing that gene, the Gus A gene, will appear blue in color. So you can visually differentiate between the tissues where your gene of interest or your promoter is can carry expression, can carry out expression, and where it can't. So in case of the first transgenics, you can see that the promoter was able to drive expression in the sepals and in anther and in stigma also, but not in the petals or in the style. So here we can expect that in native system that is in the natural Brassica gentian system, the SOC1 gene for to which this promoter belongs would be expressed in sepals and anthers, but not in petals. So this is how we functionally characterize genes and promoters. Was I able to address your query?
Yes, ma'am. Thank you so much. So it is a histochemical assay, and it is.
Yeah.
I just wanted to know that how it like what other things can we use it in for. You said that it is a histochemical assay,
Okay.
so it is used for. Can you point out some more uses of this assay?
Okay, because I'm a plant researcher, my head directly goes to plants. So I would
Botany here.
like I have used sorry, so I have used it for promoter characterization, but it is mostly used for gene characterization also, because instead of cloning promoters, you can clone genes and you can tag your reporter gene to it. So wherever your gene will be producing proteins, it will have a so even you cannot detect your gene wherever it is going in within the cell or in which tissue it is getting expressed, but you can track Gus with color.
Okay,
Due
okay,
to
that
its
is
color, you can track Gus.
yeah, the bluish color that it
Okay,
gives.
so this
Oh,
is
good.
yeah, and you can use it for fluorescence also. Instead of using Gus, if you use mug as a substrate, it will give fluorescence also. So you can track the fluorescence or you can track the this blue coloration, whichever you choose to do.
Okay, okay, thank you so much, ma'am. Any other
Yeah.
questions from other participants in the conference? Okay, then I'll take that as a no. If you have any questions, you can just type it down and we can discuss it later also. So thank you so much, ma'am.
You're
Um,
welcome. Thank you for
this,
having me.
yes, ma'am. It was a insightful
I'll stop
presentation.
sharing my screen now.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Okay.
Um, okay. Thank you, Tanu ma'am, and that was a beautiful presentation. Now, I would like to welcome our next speaker, Dr. Neha Kashyap. Skilled in genetic engineering and molecular biology field, she has worked on cloning, BIFC, pull down assay, etc. She is skilled in handling enzymology experiments, biochemical tests, and also in real-time PCR centrifugation spectrophotometer. She has skillfully grown bacterial and microbial cultures. Dr. Neha is a graduate and masters in biotech and is a PhD holder in life sciences Institute of Plant Biology. She has attended various workshops and trainings and has published papers on them. She has also received various scholarship and is skilled both in technical skills and also in the software skills. She has interests other than in biotechnology. She has interests in games like badminton, yoga, mono acting, and poetry. With um, ma'am has also attended various workshops and trainings. And with this, I would like to welcome Neha ma'am to enlighten us on the topic metabolism of lactose and tryptophan, how meticulously by E. coli genetic system. Ma'am, the stage is all yours. Please proceed.
Ma'am, please turn up your mic.
Excuse me, ma'am.
Ma'am, you're not audible actually.
You are muted. Please mute yourself.
Muted. Yes, ma'am.
Now I'm audible?
Yes,
Yes,
ma'am.
ma'am.
So I would like to thank Microbiologist Society of Jharkhand and Father Nabor Lakda, and obviously my one of the favorite, Srivastava sir, and all of the head of this organizing committee on the student, and obviously Dr. Swaranjit Singh. And today I just would like to elaborate something about the lactose operon system and how it is helpful in the genetic engineering. So I'm going to share my slide. So it's visible?
Yes, ma'am. Your screen is visible.
Okay. Oh, so today I'm talking about the lactose operon and lactose metabolism and how meticulously by E. coli genetic system and their advantages in the genetic engineering. Currently, I'm a research scholar in the National Taiwan University in Department of IPB. So my whole presentation is divided in different parts like a background introduction, research design, in which the methods and my current data and some references. Just because of Ministry of Science and Technology of Taiwan, we are not allowed to elaborate some data before publishing. So I just put some few data, not all the data. So background introduction. So before I will start my topic, I just want to remember some scientists because we always forget about that. So this Dr. Theodor Escherik. In 1884, the German microbiologist and the pediatrician Theodor Escherik discovered a fast-growing bacterium that he called bacterium E. coli. So a fast-growing bacterium that he called Bacterium coli commune, but which is now known as the biological rock star because the whole process of the genetic engineering and the molecular biology depends on the E. coli. And next, obviously, when I'm talking about the lac operon, then obviously I should remind about the Dr. Francois Jacob and the Jacques Monod. Jacques Monod, together with the Francois Chekhov, has formulated Lac operon model for the regulation of the gene expression in the late 1950s. The two of them, together with their colleague Andrei Love, were awarded with the Nobel Prize in the Physiology or Medicine in 1965. Since its discovery, Lac operon has been serving as a model system for understanding different aspects of gene regulations. So I just tell something about the E. coli, that is the Escherichia coli is known as the E. coli. It's a gram-negative facultative anaerobic rod-shaped and commonly found in the lower intestine of the warm-blooded organism. And these are the scientific name and the order and the family and the phylum. So when we talk about the genetic engineering or the Lac operon or the molecular biology, it's very important that we talk about the transcription and translation. But one of the student speaker already talked about the transcription and translation. So I just little elaborate about that. When we talk about the transcription, that the main thing just comes in our mind. That is the starting the expression of the gene. Right? There is a copying a segment of a DNA into RNA. The segments of the DNA transcribed into RNA molecules that encodes protein, which is the mRNA. And then what is the translation? That means the gene starts transcription and then it produces the mRNA, and then mRNA produces the protein. So the translation is the synthesis of the protein from an mRNA is converted into an amino acid sequence in a protein. So obviously these are the codon, and these are obviously when we talk about the DNA, these are the base pairs. So before I start the Lac operon, some useful terms I think the whole people who is here should remain in the mind to not getting confused. The first one is a cap site. The site of a DNA template where transcription begins, it corresponds to the nucleotide at the 5' end of the RNA transcript, which accepts the 7-methyl guanine cap. Second is a promoter. It is a region of a DNA upstream of a gene where relevant proteins such as the RNA polymerase and transcription factors bind to initiate transcription of the gene. The third is the RNA polymerase. Obviously, the RNA polymerase is a multi-unit enzyme that synthesizes RNA molecules for a template of a DNA through a process called the transcription. And the fourth is the operator. Operator is a region, just like a promoter. Operator is a region followed by the 3 structural genes like Z, Y, and A. And CMP, that is a CMP receptor protein, CAMP, plays a crucial role in glucose lactose by activating the major glucose transporter gene in Escherichia coli. So am I audible, right?
Yes, you are.
Yes, ma'am.
Okay. So now I'm just moving to the Lac operon, the mechanism and the regulation. So here is the whole picture of a Lac operon, but I just talk about the one by one. So this is the whole Lac operon in which there is some regulatory sequence and the operator Lac Z gene, Y and A, and here is a terminator where obviously there will be the termination of the transcription, and I will talk about that how it happens when the inducer will be present. How it happens when the inducer will be absent. So obviously the Lac operon is a well-known example of an inducible gene network that regulates the transport and metabolism of lactose in E. coli. And obviously it encodes the gene for the internalization of the extracellular lactose and then its conversion of the glucose. So the 3 important genes and their which code for the enzyme and their function. Like a LAC-Z, the enzyme is a beta galactosidase. Tanushi ma'am already talked about that. And their function is obviously in transform lactose into allolactose and also catalyzes the conversion of lactose to glucose and galactose. And LAC-Y is a gene and enzyme is a permease. The function of this enzyme is a membrane channel protein required to uptake lactose from the environment. And LAC-A, the enzyme, is a thiogalactosidase transacetylase And its function, it treats the cell of toxic thiogalactosidase, and that also transported by LAC-Y. And also here is a one more important gene or protein, that is a LAC-I, is a regulatory gene preceding the LAC operon, is responsible for producing the repressor protein. So this is the very simplified structure of the LAC operon, because here lots of students are from first year, and maybe the slides will be moved to another group where the student will be from the class 11th or 12th. So I just, and I'm also a PhD student, so I just want to remind in a very simple way. So this is the LAC operon in a simple way. It is the cap site and the promoter and the operator and the LAC-C and LAC-YC and A gene. So what happens when the glucose is present and the lactose is absent? So obviously the LAC operon consists of a promoter and the operator, and the region followed by the three structural genes. But if the LAC operon, the lactose operon of equally turned on only when lactose is available, and glucose, the preferred energy source, is absent. When there is an absence of lactose, the transcription of LAC operon gene is blocked by repressor protein. So the first one is glucose is present. That means in a media or in a media, the glucose is present, but the lactose is absent. Then what will E. coli do? What will happen with the LAC operon? Then obviously one CMP level will be low. And what will be the next? Next will be the activator cap remains inactive. When the CMP protein level will be low, then cap remains inactive. That's why it is away from the cap site. The third process will be the LAC repressor remains bound to the operator. It remains attached to the operator. And the fourth process, obviously, then there will be no transcription of the LAC operon, because RNA polymerase will not bind to the promoter, and there will be no initiation of the transcription. That means there is no expression of a gene. But it happens when the glucose is present and lactose is absent. What happens if glucose is present and lactose is also present? What happens? The first process will happen. The activator cap remains inactive because it's away from the cap site. Second will be the LAC repressor is not functional because the inducer lactose is not present. Here we can, because glucose is also present and lactose is also present. So in the same amount, we can say the inducer lactose is not fully present because glucose is over there. And here is the allolactose, which is a mimic of the lactose, is also present. Then there will be, in the condition, the basal level of transcription occurs. RNA polymerase will bind, but there is not a full transcription occurs, not a full expression of gene will occur. What is the third condition? The third condition is when the glucose is absent and lactose is absent. Then the activator cap is active as a high level of CAMP present. We can see here the cap binds to the cap site. And then LAC repressor is functional and active. 3, LAC repressor remains bound to the operator and prevent transcription. The LAC repressor will still bound to the operator, and it stops the RNA polymerase which binds to the promoter, but it stops the transcription. That means there is no transcription and there is no gene expression. But what happens when glucose is absent and lactose is present? That is the main thing in the LAC operon. The CMP level are high, so cap is active and bound to the DNA. You can see here, they bound to the cap site. And second, the cap helps efficient binding of RNA polymerase to the promoter. So this cap helps the RNA polymerase that it binds very successfully to the promoter, that is the region where the RNA polymerase can bind and is helped to start transcription. And then the LAC repressor is inactive due to the presence of inducer, lactose, or allolactose. So that means there is a transcription starts or gene expression starts. So we summarize this in a short table that you can see glucose is present and the binding of cap is absent, then lactose is absent and binding of repressor is present, that is there is a no transcription. When glucose is present, binding of cap is absent, lactose is present and binding of repressor is absent, that means a low level of transcription. And when the glucose is absent, binding of cap will occur and the lactose will be absent and the binding of repressor will be there, then there is a no transcription. But when the glucose is absent and binding of cap will be there and lactose will be present and binding of repressor will be no there, then high level of transcription, that means high level of your gene expression takes place. So how it is helpful in the genetic engineering or molecular biology, or we use it in a daily field in our molecular biology experiments. So these are some research articles. They talk about, if you are interested, then you can go through. This is the modeling network dynamic, the Lacoprona case study, some mini review, some research paper in the Nature Chemical Biology. They just tried the different sugars and just try to check how lacoprons occur. So my research design, how it helps in my daily experiment. So I just want to show to the student. So basically, I'm just working on my PhD thesis is all about with my advisor. He's actually expertise in the FINT19 gene, which is and the light signaling. So my lab is based on the hardcore genetic engineering lab. So we work on the light and the different molecular biology techniques. So my gene is my gene of interest in my research is HDACs. So basically the HDACs are grouped into 3 types. I'm just little elaborate, so you can go through with that. So HDACs are classified into 3 types, type 1, type 2, and type 3. So type 1 is the RPT3 class, and type 2 is the ST twins class, and type 3 is the seconds class. So I am focusing on the type 1 RPT3 class, that is a reduced potassium deficiency 3. It comes from the yeast cell, so that's why it's named like that. So it's also further classified into class 1, class 2, class 3, and the unclassified. So basically, right now I'm just focusing on the 19, SDA15, and the 14. And also, I'm interested in the SDA6. If any gene will, if the gene will be, my gene of interest will be further shows the interaction, then I will just move with the 1 or 2 genes. So I'm showing the result here about the SDA14. So basically, this is I'm working on the Arabidopsis. So this is the model plant, and so that's why it's the HDA19 and SDA14. Otherwise, it will be HDAC if you search in a Google. So these are the different HDAC. Like 14, you can see here the green line showing the positive regulator and the red line showing the negative regulator. So SDA14 is a negative regulator of tubulin acetylation, and the SDA15 is a positive regulator of ABA signaling and the negative regulator of photosynthesis and the chlorophyll biosynthesis. And SDA6 is a positive regulator of embryogenesis BR signaling and the negative regulator of HA signaling. And also, I was just want to check the interaction with the FIN219, and FIN219 is a expertise gene of my professor, that is the auxin regulated gene, and it also takes part in a AB signaling and J signaling. So that's why we want to check in a 19 and 6, maybe it interacts together. And the next is obviously 19, which is takes part in a positive regulator of a BR signaling and negative regulator of a ABA signaling. So I already done my biopsy data. So this is the biopsy is a protein-protein interaction. We just use this one vector, that is a PRTL2 YC and PRTL2 YN. With suppose I use the vector PRTL2 YC, that means Y C terminal. Then I need to use my gene in the N terminal. So C and N terminal, there will be a GFP, a protein will be there, tag protein will be there. So if there will be a fluorescence, then I can see, okay, my protein interact. So I already done, but I told that just because of Ministry of Science and Technology prohibition, I can't project all my data here. So right now I'm just doing on the pull down assay. So pull down assay is a protein-protein interaction. After biopsy, I'm going to do that. This is the one of the very good technique of the molecular biology or genetic engineering. So these are my gene of interest, SDA14, 15, and 19. So I cloned SDA14, 15, and 19 into GST tag PGX40-1 vector. So these kind of vectors are also available in the Google or Snapgene. You can search about that. So this is present in my lab commercially. So, and the FIN219 were cloned into CBP tag PCAL-N vector. So obviously, if one gene is in the GST tag, so next gene should be in the another tag, maybe his tag, CBP tag, or maybe MBB tag, like that. So I did so many experiments, like a check induction temperature and time, and the select high induction temperature and time, and go for sonication, and the antibiotic staining, and then pull down assay. And this is the whole process of the pull down. So this is the first, when I go for the cloning, I used a SnapGene, which is a biology software to design a construct. So it's very normal. If you go to any molecular biology lab or maybe any genetic engineering lab, they use a SnapGene or maybe some another bio softwares. So I just open it, and I just open a new DNA or RNA file, and they just open, and then I go to the TAIR. You just search the capital TAIR, that is the Arabidopsis website, where you can search your gene over there, and you can get your CDS, that is and full CDS, and the cDNA and the genomic DNA. The whole data is over there. Whole database is over there. So I searched the SDA14, and then I write the SDA14, and then I searched. And then next I get like this. So this is the annotation number. So you have to see very carefully that they have written it's a SDA14. That means it's my gene. Sometimes they have so many, and you have to search, which one. Usually the first one is basically you have to check this is your gene of interest, but you have to be very careful. And then I just click this number, and then I get the 3 or 4 tools over there. One is a CDS of SDA14, and then cDNA of SDA14, and the genomic DNA of SDA14. Then I click the CDS, and then I click the CDS. Then I got and sent to BLAST, and then I got these kind of whole base pairs of my gene is over there. So actually, when you talk about the plants genomes are very, very lengthy in comparison to animals, like 12,000, 15,000, 20,000, like that. So the cloning of a plant gene is little tough in comparison to animal gene. So I just use just paste over here in a SnapGene software, and then there is and then I just click the create a file. Then they created the file, and they just make like this kind of map. And then I go for the check the whole restriction endonuclease, because when you are going to make a construct, first you have to make in a virtual version, and then you go for the technique like in a you can say it's in a dry lab first you have to do, and then you can go for the wet lab. So, and always be careful then that when you go for the cloning, the first thing is the restriction endonuclease, the enzymes which is present in your vector, should not be present, compulsory, must not be present in your insert. Otherwise, it will cut in between the insert. Like these enzymes, I can't use. Otherwise, it will cut in between my insert. So I choose the 2 enzyme. I think I use the SAL one, and the one is EcoR1, I guess, or yeah, I use the SAL one or EcoR1, I guess, for my. So this one is the map of the vector. So here you can see here is a lac I, like I talked about the lac operon. So here is a lac I. and the LAC-IQ promoter is present over here. So LAC-I is what? It is a 3318 4400 is equal to 1003 base pair, 360 amino acid, that means the 38.6 kilodalton. We know that we just quantify the protein into dalton, not a base pair. So product is a LAC repressor, and the LAC repressor binds to the LAC promoter to inhibit the transcription in E. coli. And this inhibition can be relieved by adding lactose or IPTG. That means adding the inducers. We talk about the inducers like a glucose is present and the lactose is absent, or lactose is present. They are the inhibitors. Inducers. So this is the whole construct I made, like SJ14 PGX40-1 construct designed for the cloning. So this I made the virtually. So this is my insert SJ14 I inserted here, and I did PCR also. That's also a long process in the virtually, and design a particular primer to amplify my gene of interest. So here you can see the SJ14 forward I used EcoR1, and for the reverse I used the SAL1. And we always insert in the multiple cloning site, not any other person. That's multiple cloning site. And this is a GST tag, because I want my construct with the GST tag, because the second protein was already in the CBP tag. So both the tag, when I add the antibody, if both the tag will be interact, then I can see there will be band, and I can say, okay, my protein is now interacted with another protein. So after that, when I send for the primer, and they just send me the primer, and then I go for the PCR and did my cloning. And after the cloning, I got so many colony, like Dr. Tanushi told about that so much colonies, some are white and some are blue, but here is only white colony, but some are my colony, like my 14 PGX40-1 construct colony, and some are colony like a not transform colony. When you go for the PCR, you see nothing, like here you can see. Then I went for the PCR, and this is the PCR program, like this is the normal PCR program for the colony PCR. So I use my primer, this primer. This primer I used for my make my cloning, but here I use the GST primer. So to check about your colony PCR, you have to use the first one primer from your insert and one primer from your insert and one primer from your vector. So I use the GST primer, 5 prime, that is a forward, and one primer from the 14, my interest, gene of interest, reverse, ST14 reverse, and GST 5 prime. For this, I use the annealing temperature is 55 degrees Celsius in 13 seconds, and whole cycle is 30 cycles. So my advisor always advised that for the colony PCR, never go for more than 30 cycles. Otherwise, it shows here a very good band, but that will be non-specific band, just because of you accumulate so much gene. So that's why you can see your signal, but that was not a good signal. So here I just used the so many colony. I got like a 25 or 30 colony, and I transformed into DH5 alpha. The first I transform into DH5 alpha. Usually, apart from the gateway system, we go for the colony piece. We go for the cloning, like a biopsy and the spool down. We use a DH5 alpha. And then several colony, I randomly choose the colony 11, and some colonies are unsuccessful. You can see here, and then I choose that colony and send for sequencing to the company, and they send me the sequencing result, and I check. Oh, it was. That's my good luck. The first time it was. In the first attempt, it was correct. Easily, in the cloning, maybe you have to repeat so many times because there will be some frame shift mutations, there will be some point mutation, then you can't go for the further process because it affects your protein. So I again transform into host cell because I want my protein to express in particular. I want a particular host which express more my protein. So it's good for further pull down. So this colony, I extract the plasmid, and then I again transform it to the BL21 codon plus. This is again the E. coli bacterial cell. So there are lots of E. coli bacterial cell like a PL21 codon plus and BLDE3 also, but I successfully my BL21 shows a very good signal even in all the colony. So here also I use the 58 degrees Celsius 30-second for primer annealing. I use the same primer like a GST and the reverse. And then I pick up the colony 7, and this is my annealing temperature. And then the next step, I want to see the induction of my protein. So this bacterial colony, first, I just want to tell every student, the first make your clone successful and then store it. It's very important. So I just try to induce the protein. So my professor suggested me that it's very good to 37 degrees Celsius, but I also tried for 37 degree, 20 degree, 16 degree, and different concentration of IPTG. But I tried 0.5 millimolar and 0.1 millimolar also IPTG. So the stock will be 1 molar, and I just dilute into LB media, so and convert into 0.5 millimolar IPTG. So first, this the STF14 PGX40-1 in the host BL21 codon plus. I just in a 5 ml LB media, I just keep in a shaker for the overnight with my bacterial colony, and then after overnight, I just like a 16 hours. I think not more than 16 hours or 18 hours is good because the bacterial growth rate will be declined. So I just transfer into 250 ml, or maybe you can go for 100 ml. And then again, I keep it in a 37 degrees Celsius for 4 hours. And then after 4 hours, I just again transfer, again I add the 0.5 millimolar IPTG into LB media to see how much induction of my protein. And after adding the 0.5 millimolar IPTG, I just kept it for the 4 hour or maybe 5 hour when your OD, optical density, will be 0.3 to 0.4. But some common sense, because I work it so many times, so after 4 to 5 hours, usually it goes to the 0.3 to 0.5 OD. Then I go for the centrifugation. I just pour all the culture, like a 50 ml in the falcon tube, and just go for the centrifugation, and then collect all the cell debris and go for the sonication in the sonicator, because here after centrifugation there is a pellet, but actually it's not exactly the pellet, it's cell debris. So I want to differentiate the cell debris into supernatant and pellet. Then I just got the supernatant and pellet, and then divided it into different falcon tubes. And then I just go to check the STS page, that it's how much it is useful for me, does protein is induces or not. But before telling about my protein induces well or not, I just want to talk about the role of the LAC-I, which you have seen here, LAC-I promoter. So LAC-I promoter, so here you can see that if there is no lactose, that means lactose is absent. That means inducer is absent. So then the inhibitor protein LAC-I binds to the regulatory site of the LAC-O, which is in the promoter, right? Then there will be no gene expression. That means your gene, like my SDA40, will not express. But if the lactose is present, that means inducer is present. Then the LAC-I protein undergoes a conformational change, which changes its binding affinity for a LAC-O sequence, and the LAC-I protein comes off the LAC-O sites, and transcription of the LAC gene can occur. So there will be expression of the gene takes place. So here I didn't use the lactose. What I used the IPTG. IPTG helps in induction of my LAC-I promoter, or my LAC-I gene, or LAC-I protein. So why we use the IPTG in the molecular biology field, or how it is helpful in the LAC-O protein? So IPTG, or isopropyl beta D1 thiogalactopyronoside, is a chemical reagent mimicking allolactose. In the first 2 or 3 slides, I talked about the allolactose, how it binds, then RNA polymerase binds to the promoter, and then transcription starts. So which removes a repressor from the lac operant to induce the gene expression. So it's very important your gene should be expressed. If your gene should be expressed, then your protein will be expressed. Otherwise, there will be no expression of your gene and there will be no induction of your protein. During IPTG induction, cells must produce T7 RNA polymerases required for gene transcription. For example, you can use BL21, DE3, or BL21 codon plus. I used here codon plus. Has a LAC1 gene encoding for the LAC repressor, LAC-I. After IPTG induction, the strain expresses T7 RNA polymerase. And allolactose is an isomer of the lactose. So here in a cartoon data, in the cartoon, you can see, you can understand very nicely how IPTG induction transformed E. coli cell works and induced protein. So here you can see that when IPTG is absent, then what happens? Like when inducer is absent, like a lactose. So inside the transformed bacterial cell, if E. coli cells, in the absence of IPTG, the LAC-I binds to the LAC-O site, right? And the binding blocks the T7 RNA polymerase from initiating the transcription of your gene of interest. Like suppose there will be no IPTG, then there will be no expression of my HT14 gene. If IPTG is present, then what will happen? Adding the IPTG, I added a 0.5 millimolar IPTG. Then the IPTG enters the E. coli cell and removes the LAC-I, LAC-I releases from the LAC-O, and the resulted the T7 RNA polymerase binds to the T7 promoter and initiate gene transcription, and eventually it leads to the synthesis of protein of your interest. That means my HDA14 can express, induces. Then I can go for the pull down. So this is my some basic data that HDA14 GST. I use 37 degrees Celsius for this is the 0R and the 4R debris, and this is the after sonication. I have seen that it very high induction in the supernatant in comparison to the pellet. This is the 4R pellet. This is a 4R supernatant. Here's a 750 kilodalton, the 60 kilodalton. And we just understand the protein. We calculate the kilodalton of the protein with the protein size and also plus the GST size. So this is the whole protein. But I want to know that its protein is correct or not, because maybe it can be bacterial protein. So you have to go for the Western blot. You have to stain the antibody. If the antibody is stained, that means your protein is the correct size. Otherwise, sometimes it's a bacterial protein. So I transferred this again. I did the experiment and transferred to the PVDF membrane and just go for with the transfer buffer. I transfer to the PVDF membrane overnight and then go for the antibody staining. For the primary antibody, I use the GST B14, and secondary antibody, I use the anti-mouse monoclonal. So here after sonication, you can see here. So both the protein is correct size. Both the protein is my cloned protein. So here is in a 4R supernatant. It shows a very high accumulation of the protein, very high induction of the protein with the help of a LAC-I and the IPTG. And the 4R pellet here you can see. So that means I can go for the pull down. I can further, I can extract the protein and wash it and just use the different tag like a CBP and FIN219 for the input and the pull down, and then I can go for the pull down. But I told that just because of Ministry of Science and Technology, I cannot able to show my data of the pull down. So this is the reference and further reading like a prokaryotic gene regulation and the molecular biology of genes and the Brooke biology of microorganisms. So at last, once I was just attending a seminar and one professor from the Cambridge University, and she told that we are not only an individual organism, we are made of a group of organisms. In our gut, there is the E. coli. So we are not only one organism, we are full of so many organisms. So thank you, that's all.
Miss Aroma, are you on the loop?
Yes, sir. Hello. Just
Yeah,
1 minute for us, electricity problem here.
Oh,
it's raining also.
yeah, electricity and network, these two are big problems over here.
Always.
How is it in Taiwan? Neha?
Yeah, everything is okay, but they are so disciplined.
Oh, hyper discipline. Yeah,
Yeah,
in a
hyper
way that,
discipline.
yeah, in a way that is good. And that is required in India also to some extent.
Yeah.
So that was a wonderful lecture, and ladies and gentlemen, I hope it is a kind of our gut is already filled with E. coli, but our knowledge gut is now full with the
With E.
information
coli.
that we got from Ms. Neha Kashyap.
Yes.
Over to you, Saroma.
Dr.
Okay.
Rajesh sir,
Yeah.
Yes, sir.
please
Dr.
include
Kashyap.
all the speakers in our WhatsApp group.
Yeah, sure.
Such a
In water,
such a
in water group.
cream in biotechnology.
Yeah,
Yes, yes,
yes.
Correct. All the speakers are
Sure.
cream.
Yeah, these are gems. These are gems of today, and they will shine very bright in the future. And
Very good.
I'm sure
Very
that
good.
yes. Thank
the
you.
future
Yes.
of our coming generation will be in very safe hands with the young
Very correct. Very correct.
researchers of today.
Neha
Yeah.
ma'am, keep in touch with our microbiology society.
This is
Yeah.
sure, sir. Sorry, sir. We are great because our teachers are great.
Yes. Yes.
We learned from Indian teachers. That's so,
Correct. So that is your culture. You are maintained.
Yeah,
Yes.
it is. It is. Yeah, it is.
You are
It is.
expressing.
Yeah, it is their greatness that they acknowledge. Uh, we just tried, and we are just trying to make a platform for these young researchers.